So yes, welcome everybody to our first seminar of 2026 for the Columbia University Northern Plains Superfund Research Program. This is an exciting time now for our center. We are in year 4 of the program. This is time goes fast, especially because year 3 was a little bit accidental. So so it feels like we should be year 3 still today, but we are year 4. Nevertheless, we have lots of really amazing things going on in the center. And today we have the opportunity to learn about the great work that has been led by Project 2 of the center and the use of the isotopes as a very innovative technique for us to be able to learn more about uranium exposure and distribution in the Northern Plain communities. So with this, it's a pleasure to introduce Anirban Basu, who is the lead investigator for the project. And Anirban, I believe, he's a geochemist by training and a faculty member in the Department of Environmental Health Sciences with a very strong joint collaboration with the Lamont Doherty Earth Observatory. Anirban, I don't know if you're going to present by yourself or with somebody else, but in any case, please take it over and let us know who is presenting with you or the presentation. Thank you so much.
I'll present. I'm sharing the slides. Yeah, is the right window shared?
Yes, we can see very well.
Okay, thanks, Anna, for the introduction. So I'll present the story of uranium in Northern Plains groundwater. So I'll start with uranium, a little bit about what we know, the timeline, and then I'll talk a little bit about rocks, and then how uranium was in the rocks in the first place, and then came to groundwater, and then how we traced it. So I'll cover a wide area in these slides. Okay, so I'll start with the a timeline of uranium, and contrary to what I used to think, and I think many people think, uranium is not really quite rare. So it's among the top 50 most abundant element in Earth's crust. It's more abundant than gold, silver, tin, and some biological essential elements like iodine and selenium, which we need. And also some elements that we are very familiar with, like arsenic or cadmium, mercury. Uranium is more abundant than all of these. And uranium is used for a long time, although the use changed quite a lot with time. So this photo is of a Roman tiles from 79 CE. And this greenish glass used in the tiles had about 1%, 1 weight percent uranium. So Romans used uranium to color glass. But I'm quite sure that they didn't know what it was back then. And this is the timeline of uranium since its discovery. It was discovered in the same year as when the French Revolution started by a German chemist, and then was isolated in its pure elemental form in 1841. But then up until 1896, this radioactivity was not discovered. Radioactivity was discovered by Becquerel in 1896. Prior to this period, uranium is mostly used in coloring glass and ceramic, and some interesting application of dental implants and things like that. But soon after discovery of radioactivity, when radium was discovered, then there was this search started for uranium because radium had its use in medical applications for isotopes. and that was the beginning of the first uranium rush. And then nuclear fission was discovered in 1938, and this is when other applications of uranium, particularly in weapons and military use, and also later on in energy sector, started really in 1938. And this initiated the second uranium rush. And 1956 was basically the first reactors in the US. Um, and around that time, it opened the doors to use of uranium as an energy fuel, really. And then the third uranium rush coincides with the oil crisis peak in 1973. And we are probably going to see a fourth uranium rush soon. So, and interestingly, I'll talk about it later, but this is very similar timeline for groundwater use. But anyway, I'll come back to that later. And if you look at the health effects of uranium, because it's been used for a long time for various reasons, the very first animal studies on health effects date back to actually 1824. So this is a cover page of a study by Gmelin, and it looked, among other elements, uranium, the effects of uranium, that's what it says in German, I think, on organisms. And the first studies that used systematically uranium in animal studies was done around 1851. And it did find very definitive demonstration of renal effects of uranium. And from that point onwards, there were two different aspects of its use. One was, very interestingly, perhaps misguided research as a treatment of diabetes of uranium. So this is a BMJ paper from 1895, where this person uses uranium nitrate to treat diabetes in humans. And then this is a commercial of a wine, uranium spiked wine, sold as a cure for diabetes. But the point here is that as early as 1850s, it was known that uranium caused renal effects. And some studies identified it as a purely tubular poison. That's what this German sentence says. So it affects the proximal tubule and causes toxicity in proximal tubule cells in the kidney. So it was known then, and fast forwarded to now, we know that yes, primary target organ of uranium is bones and kidneys. And then also it affects neurological systems, lungs, livers. But the source of uranium or exposure of uranium happens via mainly inhalation in certain settings, but more widely through ingestion of uranium contaminated groundwater and also uranium contaminated food. And to some extent, not too often, through dermal contact. And our recent work done by our broader team shows link to chronic uranium exposure through drinking water with chronic kidney disease risks and uranium exposure association with kidney tubule biomarkers. So this is what we know over last, so 200 years, more than 100 years of research. Uranium accumulates in kidneys and it causes renal damage. And the EPA drinking water limit is 30 micrograms per liter with a goal, that's the maximum contaminant level goal of 0, which means that there is probably no safe concentration of uranium. Now, the exposure comes mainly through groundwater, and it makes sense if you look at where uranium is in this map. This is a map from the Department of Energy, and I took this from a report that was submitted to US Congress in 2014. It shows the number of mines and total uranium ore production in tons and this in western half of the United States. And if you look at this pattern, so higher ores were produced where you have uranium and higher quantities, of course. And if you look at this distribution pattern with where the major aquifers are, then they kind of coincide. So the layers with high uranium, the geological layers with high uranium, are the same layers that are the aquifers in these regions. Particularly for this study, I'm going to focus on the northern plains in this part, and it's a different aquifer, but then Great High Plains aquifer and Colorado Plateau aquifers are the ones, they are dominant aquifers here with highest probably levels of uranium anywhere in the world. So this groundwater uranium is reflected in drinking water uranium, and this is a map from Ravalli et al. Paper. This study was led by Annie Nigra, which shows uranium concentrations in community water systems. We do have a good map for this, but what we do not have is a really comprehensive map of uranium distribution in groundwater from this entire Western United States. I just want to say that two Superfund trainees, Orijit and Ellen, they're starting to work on creating a map in groundwater of uranium distribution in this area. So groundwater is a major exposure source, and uranium comes to groundwater from the rock. So let's take a quick look at what setting of rocks are there in the Northern Plains aquifers and how uranium came into them. So now when I'm talking about rocks, it's useful to go back to about 100 million years from now, when the United States looked completely different. There was a major, massive water body in the middle, and this would be the South Dakota right here. And this is called the Western Interior Seaway, where a shale layer was deposited, which is really the floor of the aquifer system in Northern Plains, particularly in the tribal land, OST land that we are focusing on for this talk. So this is the floor of the aquifer. It's impermeable. That's the base of the aquifer. And over it, fast forward to about 35 million years ago, it looked quite different. The difference is that now we have the uplift of Rocky Mountains and the Black Hills. So they started eroding. So rivers actually got sediments from those areas and started depositing the sand layers, as rivers do. And this was interrupted by repeated volcanic ash fallouts from the volcanoes that existed western part of the United States back then. So this was a setting where these layers combined, collectively called the White River Group, was deposited. And these layers are mainly sand layers, but then it had interlayers of volcanic ash, which really was volcanic ash back then, but now they're weathered to clay layers. And these are the source of uranium in these sand layers, which, in some cases, they are also the aquifers. So the wells are drilled within these layers in some cases. And we know that the basal part hosts some ore-grade deposits. And then the ash layers were sourced from volcanoes. So this is one example here that shows the Southern Basin and Range volcanic field between 36 to 18 million years ago. And the wind direction was such that the volcanic ash was deposited in this area here, where the White River Formation was being deposited. Now, volcanic ash naturally has a lot of uranium. Uranium doesn't fit in any other rock-forming mineral, so it kind of hangs around in the volcanic ash. And then when it's deposited, then the dissolved oxygen in groundwater, when it comes in contact with groundwater, it oxidizes and solubilizes uranium, makes it very, very mobile, and moves it through the other layers, particularly the layers below it. So that's really the story of uranium, the origin of uranium in these layers. And then this White River groups, above it is the main aquifer, which was deposited afterwards, climate changed slightly, you know, from the White River time, but then it was very similar deposit, mostly by rivers and interlaid with volcanic ash. So ash deposition continued through it. So this is the main aquifer now, the Arikari aquifer, and in some cases, White River aquifer. But then if you go to this area in South Dakota, what you'll see is the thickness of these layers are not uniform everywhere. So that's why it's useful to look at the maps. In some cases, the thickness of Arikari aquifer actually decreases as you go from south to the north. So that's obvious kind of in this map. So now you're looking at a geological map, and here this reddish brown or purplish color, that's, you know, most of this area with the symbol TA is the Arikari formation. That's the main aquifer. So if you have a private well, very likely that you are drilling within this layer. But then as you go to the north, you have this layer that kind of at the surface. So the thickness of this Arikari layer decreases, and then you have the contact here with the White River group that is known to host uranium deposit. But we don't really know where those deposits are. The big ones were mapped, but the small ones could be anywhere, because this uranium mobilization is going on for a long time. So that brings us finally to private wells, where uranium shows up in water. And this is known for a while now that these private well waters have a lot of uranium. This is a study from 2021. One of our supercontinent trainee, Marissa Sobel, published it. And it's the same study setting, this OST land, where, you know, we have this White River group in the northern part, and then Arikari aquifer, now a different color, this bluish, this is, and then each dot is a private well. Now, the color coding is such that red is the wells where both arsenic and uranium exceeds the MCL, and black is where uranium exceeds the MCL. So there are some hotspots there. But at this point, we don't know where uranium is coming from, particularly. We know that it's showing up in the wells, and we don't know what controls these hotspots. So that's where we started our project, and the cycling is quite simple. When uranium is oxidized, it becomes extremely water soluble. And when uranium is reduced, it becomes insoluble. So it kind of falls off of the, precipitates out of groundwater, and then forms the deposits that look like this in this two figures. So these are, in the reduced areas, uranium forms these kinds of deposits. Then when condition changes, these deposits, when exposed to oxygenated groundwater, they can oxidize, and then you'll have, again, release of uranium in water. So it's really important to understand where oxidation and mobilization is happening and where the opposite is happening, that's taking uranium out. So that's one of the major questions that we had. And also for the community, it's incredibly useful. And this is actually led by the community, this particular question. Where is uranium coming from? Is it coming from a nearby source? Is it coming from a historical source far away? And we wanted to understand the same thing as well. And then we wanted to understand if we can trace uranium not only in water, but also after you drink water, where uranium is going to. So all of these questions we addressed using a very novel application of uranium isotopic tracing. And for these slides, I'm going to present the work that's done by Dr. Orija Benito, our postdoctoral trainee. So he did, the entire heavy lifting here, from sample collection to analysis to data analysis, everything basically. So we are using two different isotopic tracers, one to understand the cycling, the other one to understand where it's coming from. And the first one to understand the cycling, this tracer we are using is the ratio of 238 to 235 uranium. So natural uranium has three isotopes, 238, 235, and 234. So we're using the ratio of 238 to 235, which is uniquely sensitive to the cycling. So what happens is this theory was proposed by Bigelaisen in 1996. So it's fairly not too old. Um, and the idea is that when soluble uranium is reduced in insoluble uranium, that favors 238 uranium in the insoluble part. So if you look at the water becomes enriched in uranium 235. So this ratio changes in a way that if you measure the isotopic proportions, then you'll have water that has more 235 compared to 238 if uranium is being removed from water. Opposite happens when uranium is released into water. So you'll have then more 238. And this has to do with a very interesting, very fundamental property of uranium, that its mass, as the mass increases of this isotope, the volume doesn't proportionally increase with the mass. So what that means is uranium-235 is smaller than it should be. So that's basically what causes this very interesting effect. And this effect is measurable. Really, we can quantify it very, very well from water samples. We're looking at these water samples from the groundwater wells, and we know that we have studies where, if you reduce uranium and remove uranium from water by bacteria, and these are the batch reactor experiments in the lab, where uranium is reduced by bacteria, so uranium concentration going down in solution, bacterial media, and with time. And if you look at if you follow the isotopic composition of the same water, then you see the isotopic shift is very clear, very systematic. And I'm going to present the data in delta 238. So this is really the ratio of 238 to 235 normalized to a standard. The way to read it, if it's 0, nothing happens. If it's negative, 235 is more, 235 enrichment. If it's positive, 238 enrichment. And it's not unique to microbes, although these microbes are the main reason why uranium precipitates in water. Iron-reducing bacteria does the job. But then also a biotic reaction does the same thing. So the bottom line is that we can use this isotopic tracer very accurately because it's sensitive to uranium reduction and nothing else. So that's basic, the fundamental science behind it. And then we are using another isotope ratio, which is 234 to 238. So this is where the radioactivity part of uranium comes into play. And we have 234 being produced from the decay of uranium-238. Now, 234 is also radioactive, so it decays as well. Now, with time, the way it works is the production of 234 is balanced by the decay of 234. And that balance is called secular equilibrium, where the ratio of these two, when normalized by the decay constant, becomes 1. That's quite useful to see, that when that value is 1, that means it's in secular equilibrium. Now, what's very interesting here is when 238 uranium decays, it spits out an alpha particle, and it's just like a recoil of a gun. The daughter nuclide is recoiled by a distance of about 30 to 40 nanometers. And this creates a huge difference because if that uranium atom is sitting in a uranium mineral grain right at the boundary, then it can be kicked out of the grain. And when it's kicked out of the grain, it goes in the water and the water takes it away. So the end result of this, if it goes on over many, many, many years, thousands, hundreds of thousands to millions of years, what happens is the uranium ore or the uranium deposits that are forming in these geologic layers, they develop a very characteristic isotope ratio. That can be used to trace if these things are being mobilized in water. So usually these uranium deposits have a ratio of close to 1. It could be even from 1 to 1.8 in that range. Now, this is useful to see in the cross section here. So this is actually for the yellow bit, the uranium deposit. If your well is here, then the water that's not getting uranium from this deposit will have an activity ratio much higher than 1. But if there is mixing with this, then it can have activity ratio of close to 1. So basically, these things have very unique characters. That's in the range of 1 to 1.8 usually. We use that to trace. Now, if it's traveling for a long distance, the decay will increase this ratio. And so higher the ratio, that means higher the travel distance or longer the travel distance. And that's useful to see, if it's from a nearby source, then the ratio is going to be low. If it's from a distant sources, the ratio is going to be high. So that's the basic concept. Just to remind you, we were looking at why the hotspots are there, what's controlling them, and where uranium is coming from. And we did the sampling with our incredible field team put together by Missouri Breaks. We sampled there, and then we brought the samples back to Colombia, where we did at Lamont some processing of these samples, and then we measured 2 different isotope ratios. And then we actually plotted our results on a map to show the spatial relationships. And this is really interesting. When you plot it that way, you see if the hotspots give you a particular pattern in terms of cycling. So one isotopic composition, that's the 238, 235, that gives us cycling where uranium is being released. And then the other one, the source proximity. So that's basically the idea. And again, this whole thing was done by Orijit. So for this one, I'll start with the concentration pattern. This is the map of South Dakota. Our study site is here. And this map shows the concentration distribution in water. Now, to orient you, the water flow is from actually the general direction of water flow is from south to north, right? So it's flowing from south to north. What we see is we have a hotspot here where the concentration goes above the EPA-MCL. And we tested about 140 private well samples. All had uranium, detectable range, but only 5% were above the EPA-MCL of 30 micrograms per liter. So there is this hotspot here. There is one high concentration here, one high concentration here. But it doesn't tell us where the cycling is happening, where it's oxidized, where it's reduced. And that's where isoscapes, the spatial distribution of isotope ratios in water, comes very handy. So basically, this is the 238 to 235 isoscape. This is a cycling isoscape. So this big red hotspot, the red values, the way to read this map is, again, we're looking at 238 to 235 ratio here in this value. So anything above, say, 0 is the signature of uranium release under oxidizing environment. This dark blue, there is no way you can generate these types of numbers in water samples if there is no uranium removal by reduction is going on. So what we see is, except for this one hotspot, this zone 2 is quite reduced. So in this zone, uranium is being removed from the water, and in this zone, uranium is being added to the water. So that's very helpful. It tells us the cycling, that this is very likely, if nothing is changed, then probably this trend is going to remain the same. Maybe, it can get worse. But if everything is left the same, hopefully with time, because the reactions in the right direction, uranium is going to be removed from water in this zone, except for this one hotspot where also we see dissolution is going on. Now, what's really striking is this other isoscape of source proximity, where this brown color shows you proximity to the source. So basically 1.5 to 1.8 in this range, closer to the source. And then green, this very high values, means that uranium has to travel a long distance or for a much longer time with groundwater to get to the well. So that means it's coming from a distant source. And this is very interesting and really helpful, because if I put these two maps next to each other, if you remember, I mentioned that most uranium is in this brown color geologic deposit. So you're looking at a surface map. And as the wells are drilled within Arikari Aquifer, as you go from south to north, it's more likely to encounter this layer with high uranium, high frequencies of uranium deposits um, closer or at less depth as you go north. So what we are seeing, particularly here in these wells, we are seeing uranium that's very likely at least it looks like, coming from the White River Group rocks. So this is so the source of uranium is also, as you see, the high concentrations here, and also in our map we show high concentration, the same pattern here. But then source is lying closer to the surface in that geologic formation, White River Group formation. That is the problem. So this is really very helpful up to this point. And then we also wanted to understand that if this story that we are getting from uranium isotope, if that checks with everything else that we collected, for example, iron concentration, manganese concentration, a whole bunch of other parameters that tells us about groundwater chemistry, also what kind of bacterial reactions are going on. So when we did a clustering, we see basically 3 clusters, but then it's not super separated. There is an overlap, and usually to visualize the structure in the data, normally people use principal component analysis, but that works really well for linear data. What we are seeing here, what we are using, is one sample with about 15 variables, where the variables have a wide range. So they're changing quite a lot. So very nonlinear data. So we are using this new approach in groundwater chemistry, uniform manifold approximation projection, which preserves the local structure and the overall global structure in the data, and shows and handles nonlinear data really, really well, much, much better than the PCA. So you plot the same data with the PCA, you wouldn't see this kind of structure. So UMAP shows 3 clusters, spaced differently, but there is also an overlap. So gradual change from one cluster to another cluster. And when we looked at the box plot, of the uranium concentrations, isotope ratios inside these clusters, and that matches very well with our redox story, which really is controlling the uranium oxidation or reduction. So blue is the oxidizing area, so northeastern hotspots where we have highest uranium concentrations we have observed. Orange is the reducing part, where we have observed the most reducing or most negative delta values, so 235 enrichment, and the green is basically mixed. And when you look at the z-scores of major analytes, for example, here we have selenium and vanadium, which would also increase if the zones are oxidizing, and iron and manganese will increase if the zones are reducing, because these are the products of microbial reduction, and the same microbial reduction reduces and precipitates uranium from water. So cluster 2, the reducing part, shows high iron and manganese, and cluster 1 shows high selenium, vanadium, and arsenate concentrations, showing basically oxidation in these. So this all matches really, really nicely, actually super, in a way that's super well behaved in the overall data.
Can I ask
And
about
when
the electrical
yeah,
conductivity? Because that also seems to have a pattern. Yeah.
um,
That,
electrical,
that is higher in the cluster 2 and negative in the others.
yes. This we are not sure, because electrical conductivity is a combination of a whole bunch of other salts. So this is there is a pattern definitely, but it could be from several things. So we didn't focus on that, but
Okay.
we'll
Sounds
maybe
good.
we should look more into it. So when you look at the spatial pattern, so on the left, the top one shows the manganese concentration. So the way the reduction works in groundwater aquifers, you have first oxygen dissolved, So aerobic bacteria would use that, and then you'd have a zone of nitrate-reducing bacteria, and then iron. So it's more progressively reducing condition. At some point, iron will show up. But before that, manganese shows up. And these are bacteria literally breathing the solid rock. So iron is released from the rock to water. And the same reaction kills uranium in water. So this is really interesting that when you look at the reduced zone, we see high manganese, and in here, high iron, dissolved iron in water. And if you look at selenium concentration, so this top right is selenium. First of all, there is not much selenium, dissolved selenium in. So MCl for selenium is 50 for total selenium. So selenium is not a problem in this part of the northern plains, not a problem at all. That's good news. So there is this reduced zone where we have almost no selenium. So highly reduced areas completely killed all selenium from water. So selenium, when reduced from selenium 6 to 4, it kind of sticks to surfaces. And then when it's reduced to selenium 0, that's bright red selenium, which is insoluble. Something like that is happening in the reduced area. On the other hand, where we have high uranium, we have really high selenium. And this one is vanadium, which follows very similar redox as uranium. And we have the exact same pattern. This is like high, almost exact same pattern. High vanadium coincides with high uranium areas. So everything else really nicely checks out, with the interpretation that we get just from the isotope story. So the summary is that the isoscapes so far, what we have shown you, is super useful to understand where the uranium is coming from rock to water, or is it going back from water to rock. That's very clearly the siting can be identified, which is only unique to uranium reduction. So, for example, if you have a lot of iron, that doesn't mean uranium reduction is happening. There is a whole range, and the reaction can be quite slow. Only following uranium isotopes definitively demonstrates that it is a result of uranium removal from water. No other parameter can give you the same information. So then the important thing is the activity ratios show a source proximity, which is really, we demonstrate that uranium is very likely coming from the White River group, because we do know that that rock layer hosts uranium, but we don't know where and how much. This first time shows us that it is coming indeed from those areas, and those areas probably have high concentrations in the rock. So then the take-home message is that, yes, we have very sparse data, but this type of information is super helpful for the community, that now we can have targeted resampling. We know now where to sample it. Our goal is to make a time series of this isoscape to see the evolution. But with this data, we know where to look and where to focus. And where you have limited resources, that's super useful. Very targeted sampling and monitoring. So that's this part. And now the last part, that can we use, now we are very ambitious, can we use the same isotopic tracers to tell us something useful about uranium exposure to humans? Because when people drink this water, they're exposed to uranium. I have shown you that it's known for a while that uranium goes to kidneys. Can we say something meaningful about that? Now, if you look at the uranium studies, most of them look at, when it comes to health outcomes, bone and kidney toxicity, because these are two major organs. And then most of those studies actually look at protein interaction after absorption of uranium, what proteins are taking up uranium in the human body, and of course, the effect of oxidative stress after uranium ingestion. And what we know is uranium is particularly good at combining with iron transport proteins after absorption. So it's absorbed after you drink it or eat it. It's absorbed in the GI tract. And then in plasma, it combines very effectively with iron transport proteins and calcium transport proteins. So this is known. And those proteins deliver uranium to kidneys and bones. Now, is there anything that tells us how much uranium is in the kidneys? There is no easy test available for that. Kidney biopsies are very complicated, not done as a routine thing. There is nothing that tells us how much uranium is in the kidneys if someone is chronically exposed to uranium for a while. And something that tells us about that in urine, that will be the answer to this problem. So we would know then by looking at urinary markers how much uranium is stored in the kidneys or in bones, and then of course if that initiate kidney damage, then we can have some assessment of that as well. But right now there is nothing. So that's where we are using isotopes, because we are looking at drinking water anyway from groundwater. So we have the isoscapes. So our goal was, if after ingestion, body takes up one isotope over the other preferentially, then urine is going to have a different isotopic composition compared to the drinking water. And that difference is going to give you how much uranium is stored in the body. So that was basically the hypothesis. So we started from there, and this part of the study, all the heavy lifting was done by Catherine Lucy. She did a remarkable job in running the experiments, and the experiments were designed as a collaboration with Project 4 of our Superfund. So in this study, we wanted to ground truth our hypothesis and see whether it really happens, whether this hypothesis at all works. So we exposed a group of mice to 50 milligrams per liter water uranium in 3 different exposure groups for 2 days, for a week, for 2 weeks. And then we stopped giving them uranium. We just gave them uranium-free water with everything else the same, but no uranium. And then there were 2 groups, and we are calling them washout groups. And then we, after the experiment was done, the mice were humanely euthanized, and then we looked at their organs for how much uranium was accumulating and the isotopic composition. And our hypothesis is this, that we know from some experimental studies that all that strange, interesting effect of uranium isotopic behavior works in a very different way. So unlike cycling, there is no redox for uranium in organs. It's all oxidized always. But in this case, what it does is stronger bonding environments preferentially takes up lighter isotopes. That's how it works. So if that's true, then proteins will take up lighter isotopes. They'll deliver that to the organs. So the organs systematically will be isotopically lighter. So that was our hypothesis. Lighter means 235 uranium enriched. So if you look at the data that we got, and these plots show you how uranium accumulation happened in kidneys and bones. So this is micrograms of uranium per gram of tissue. And relative to control group, we saw increase in uranium with continued exposure. So with higher exposure time, more uranium. And then during washout time, what happened was that stored uranium was actually released, right? So they excreted the uranium, both from bones and from kidneys. Now, if you look at the isotopic composition, that's the plot on the right side, the way to read it is, if nothing is happening, then everything should fall on this purple bar, because that's the drinking water isotopic composition. Now, if organs are taking up preferentially 235, then these numbers should be negative. So what we see is GI tract and feces, they're very, very close. They're almost the same as the input drinking water. What that means is that GI tract and feces accounts for almost 99% of uranium, right? And that's upstream, basically, of the absorption. Now, when uranium is absorbed in gastrointestinal tract, that's the downstream of absorption. That's these 3. So basically 2 organs and urine. So what we see is kidneys are isotopically really light. So our hypothesis was validated that kidneys are taking up light isotopic compositions. Kidneys are preferentially taking up 235 uranium. But relative to 235 uranium, what's being released is 238 enriched uranium in the urine. So this, for the first time, we showed that this happens in animals. And our working explanation is this, that if you look at the bonding environment, and this is from a modeling study of uranium in transferrin, that's the iron transfer protein in plasma. So the final product has actually shorter and stronger bonds than the initial uranium compound that's in something like in plasma. So this systematic take-up and binding with protein favors the 235 enrichment. So this actually, the energetics are worked out in this paper very well. So that is very consistent with what we are observing. Now, if this is true, then once, if you look at the washout experiments, that the animals, their kidneys took up 235 uranium for a while, and then we stopped the supply of uranium. So now body is releasing that uranium, but it's releasing 238 preferentially. So what that makes is what's left there, what's still bound to the kidneys, are really, really isotopically light. So even we started from a 235 enriched pool, that becomes even more 235 enriched. So this axis is that the scale here is 3 times more, a factor of 3 more than what I showed you in the other one. Now, this is really the first demonstration of this actually happens in animals at organ levels. And this, what we show, that as the kidneys are isotopically different than urine, this uptake in kidneys is reflected in isotopic composition of urine. Now, this is the first step to develop anything that could be used as a biomarker. And this paper was, so the NIHs liked this paper. This was selected as the paper of the month for NIHs. And all this work was done by, the groundwater work was done by Orijit. The mice work was done by Catherine. And of course, this mice work was done in collaboration with Project 4. And none of this work could have been done without Missouri Break's help and their knowledge and their putting together the team, local team, and then of course all projects in the Superfund. And I leave you with this quote. This is a quote of a Lakota elder that I got from when I attended the OSC technical team meeting in October. And it kind of stayed with me. And I was like, this is really, this kind of tells us we need to take this type of work really seriously and what it means for the communities. So with this, I'm happy to take any questions.
Thank you so much. And the quote was very powerful. So thank you for bringing it at the end. Amazing work. Questions, comments for Anirban? I have a first question myself.
Okay.
This is related to with the work, what you showed from the distal sources versus the nearby sources. You mentioned that it seems that the White River would be like where it's coming from. So my question is do you need samples from the White River?
No. So
would that help? Can you do isotope measures from in the White River or even in the sediments, soil? Does that make any sense, or that doesn't make any sense?
No. So there was a miscommunication here. So what I meant is White River Group. That's what the rock layers are named. So
Okay.
they are collectively called White River Group because the White River was probably the one that deposited them over many millions of years. But the actual White River itself can have only very limited influence because the flow direction is the opposite.
Okay.
So the southern part of the study area is elevated. So that's high, and then White River is low. So the groundwater drains toward White River. So if anything, it'll push probably. So, but there is, of course, groundwater surface water interaction. So some of the effect would be restricted to shallow parts that's very close to the White River. But as it happens, that as you go in that area, the top layer disappears. So what you have even on the surface is really that White River group. And then with depth, so if you are drilling a private well in the northern part, you're more likely to get White River group at a shallower depth. So you're more likely to encounter uranium-bearing region at a shallower depth than the southern part of the study area. So that's why it shows the same concentration pattern, and the activity ratio patterns match really well with that.
Okay. Very good. Thank you. And I see a question from Sarah. Sarah, thank you.
Hiya. Yeah, thank you very much for the talk. I thought it was really good and I've learned so much. Um, but yeah, I'm curious to know a bit more about like your theories on how much influence perhaps the microbes might have with the zones of where you see, for example, more uranium reduction in the Pine Ridge Reservation. Or do you think
I
that's,
was, I,
due to abiotic
I don't think it's
processes?
abiotic unless you have very, like, my understanding is that if you have sulfate reducing environment where you have really, iron sulfide precipitation, and those are not very common in most part of the aquifer here. There can be microenvironments, but I think it's mostly um, microbial. The reduction, I would be surprised if there is any, abiotic reduction. I mean, if there is any, then it could be that it's just iron 2 minerals absorbing some of the uranium and reducing it on the surface. But the extent to that, I would expect to be really low. And then what we see very clearly is, as the manganese concentration in water increases and then iron concentration increases, those are the areas where we see really um, um, uh, negative delta values. Now, there are some studies that show that aqueous iron 2 can reduce uranium, but it's kinetically really slow. So uranium
Yeah.
reduction happens quite efficiently when there is a surface there. So that's why I think even if the abiotic component is there, it's very much microbially mediated because those iron 2 minerals were probably the result of precipitation of what iron 2 was available there.
Okay.
Um,
Well,
yeah.
that's
So
encouraging. I didn't really introduce myself, but I'm a PhD student from the University of Bristol. And a few 2 years ago, um, Ben Bostic, which I think I saw he was present here today, he kind of invited me to come to the Pine Ridge Reservation, and we collected some samples, and I've done some DNA extractions on them. So I've done some 16S sequencing, and I've also just been doing metagenomic data on the water. So I'm starting to look at the taxonomy of basically iron reducing bacteria, which can also reduce uranium. So it'll be really
so you're
cool.
the one who is going to tell us what's really happening,
Well,
whether I'm right.
we didn't collect that many samples, but so probably won't be able to definitively say anything, but yeah, it might indicate something.
It'll be
But it
really
was really
informative.
cool
I
to see
mean,
your isotope data because it's kind of following some interesting patterns to what I'm seeing with taxonomy and the gene
And the
function.
thanks.
samples, Sarah, the samples are the same or similar, at least similar locations. Do you know if they are the same households or the same areas?
Yeah, I think so. Kind of. I only got 12 samples throughout the Pine Ridge. And I think Ben planned for us to visit sites that are high in uranium and arsenic. So we have, yeah. I think I was having a look at your coordinates, I think from your SI to see if they matched up with mine. And they're not exact, but a few of them are quite close to each other.
Mhm.
So I can kind of use that.
I think
So if you need any help, sorry, Anna,
no, please
sorry,
go
go ahead.
ahead. Yeah.
So if you need any help with the coordinates, I'm going to volunteer Orijit now here because he has all the coordinates and everything in a very cleaned up version. At least he knows where the most cleaned up things are. So we, if we are of any help, please use whatever Orijit has. We're very happy to share information with you.
Okay, yeah, I'll get in contact with them. Yeah, thank you very much.
Thank you. And it would be wonderful for you and your team, to present, to come. We would be delighted whenever you feel ready, that you've analyzed the data, you understand it really well. You can come and present in the next coming months or whenever you feel ready. I think it would be really wonderful.
Oh yeah, I'm sure we'd love to do that. Yeah, I'll get in contact with you. I think I've got your emails and everything.
Mhm.
I have questions.
Yeah.
Please, yes, thank you for joining us.
Right. Very informative presentation. Thank you. I have a couple of questions. My first question is regarding the trace the uranium resource. So you mentioned the ratio of 234 over 238. So my understanding is 238 is about 99% and the 234 is tiny amount. So how did you get a ratio about 1? Similarly, when you mention 234 versus 238, so those two are in different decade chain. Does this make sense to look at this ratio? My second question is regarding the biomarker. This is always a challenge when we would do the epidemiological research. So urine, my understanding is, urine is right now is most widely used biomarker for uranium. The environmental half-life for uranium is almost infinity. But for urine, the biological half-life is kind of short. As the sun shows like days or a couple of weeks. People, very few people, live in the same place for the lifetime. So people always moving around. How can we use this urine biomarker to trace, the lifetime exposure?
Thanks. Great question. The first question is I actually left out some of the details. The raw ratio of 234 to 238 is not 1. It's very tiny. So we actually use a very special device in the mass spec. It's called an ion counter, because it's 0.005% abundance 234 compared to the whole.
I see.
So we measured them very accurately, very precisely. And then when we express it, we normalize the ratio by their decay constants. So when you normalize it by their decay constant, what happens is the natural abundance how much of 234 you would see is going to be linked to the decay constant. So the stuff that decays faster is going to be less. Stuff that decays for a long time, that's going to accumulate more. So we use that to get to this kind of dating technique, which tells us that how long it takes for something to get to that ratio of 1, because that ratio will be a long-term result of this decay of both 234 and 238. Because of the differences in decay constants, 238 is huge, which is 4.6 billion years, as long as the age of the Earth, and 234 is 245,000 years. So compared to that, it's really fast, right? So then what happens, that balance means a certain time where the production of 234 decays, the production of 234 is balanced by the decay of 234. So that is quite useful to see the geologic age. And this particular deposits, they have their unique characteristics. So that's what is helpful for us, that we can kind of pick up the signal that's coming from these deposits. And these are almost stable isotopes for human lifetime. So we can use this signal. The second question, I think it's very challenging. What we can do, what we are limited by is the amount of uranium that's in the urine. So if you look at the median value of the, say, MESA cohort or strong heart study cohort, strong study's median value is much higher, but it's at the PPT level. So we need a lot of urine. So we need 24-hour urine. So if we have 24-hour urine, then we can do it. But it's not going to be lifetime exposure, because what happens is the 70% of uranium that's ingested is excreted in urine daily. But then the rest, that kind of either is in the kidney or is in recirculation, that amount will have its effect on the isotopic composition of the urine. So it's really a long-term steady state kind of balance, where we should be able to see how different compartments, so kidneys, are in a steady state with urine at that kind of time scale. We won't be, I'm not sure if we would be able to do lifetime exposure. Maybe, if we can find enough in teeth, that may be another way to look at lifetime exposure. But I don't really know how much you need to expect.
Some cohort study collected the multiple times urine samples. I wonder, is there any way to see the correlations, see the difference one time, twice or multiple times, how their measurement is diluted or, improved?
We do have the data, actually. We have repeated samples. And actually, we can see that there has been a decline, which is related to the switching of the drinking water in the communities. And the data from the Strong Heart study has shown that before there was the switch in the drinking water, the levels of the metals that were mostly coming from water, such as arsenic, uranium, were actually pretty stable over a period of 10, 20 years. We have data showing, even if people change their behaviors, because the water source was so stable and the participants live in that area, they didn't really leave the area, the levels were very stable. But that has changed since 2006, 2009. In the last 10 years, 15 years, things have declined, and we can show that in our data. There has been an important decline in recent years. Yeah, but the repeated samples are going to be very helpful to look at the health outcomes. And that's what we are now working on. We just got the data, has been very recent. And so we are hoping to be able to move that forward now. I think we are at time now. It's after 5 minutes after the hour. So thank you so much, Anirban and team, for the incredible work I believe this webinar is fantastic and is going to be really useful for anybody interested in uranium. The historical perspective that you gave us was really phenomenal, not just in uranium itself, but also the geological conditions in the northern plains of why uranium is so important in that area, as well as the novel biomarkers and the novel isotopic tracers that are very relevant for the communities. So thank you, and thank you everybody for joining us. Next month will be in person, we hope, or hybrid. We hope to be hybrid, and we are going to switch to health outcomes. We'll be looking at brain health outcomes related to metal exposure, among other things. So thank you so much. Bye-bye.
Thank you, everyone, for being here, and thanks
Thank
for
you.
this opportunity.
Thank you.
Well done. Thanks.